Bismillahirrahmanirrahim. Assalamu alaikum. Hello and welcome to Bioscience with ShahTareen Swati. And I'm here to deliver my third lecture from the biotechnology series. And as the title indicated that I'm going to discuss the genetic engineering. In my previous lecture, I discussed in detail about the DNA sequencing, especially for Sanger method I discussed. So do subscribe my channel, like my videos, share my videos. If you're sharing my videos, you're sharing the knowledge. So keep watching, keep sharing knowledge with other. So now I'm going to start my genetic engineering lecture. As we are familiar with the genetic engineering is the transferring and cutting of the gene from one organism and then rejoining and making the new gene, or you can say that the recombinant DNA. So in this lecture, I will discuss in detail about the gene cloning, recombinant DNA technology, and many more. So, as you can see here, the outline of the lecture, I will just give you the introduction about the gene cloning, then recombinant DNA technology, recombinant DNA, a little bit about the histories and restriction enzymes. I will discuss restriction enzyme in detail, about the history of the restriction enzymes, and many more in my next lecture. So here I will give you some information about the restriction enzyme, about the cloning vector, and then steps of the recombinant DNA technology. So I'm not wasting my time. So all the students must hold on your notebook and pen and pencil and note down all the important information related to the recombinant DNA technology and gene cloning. So what is the clone? Clone is genetically exact copy. This describes the process of copying fragments of DNA, which can then be used for many different purposes. Like we use the using genetically modified GM crops, genetically GM for genetically modified crops, animals are finding a cure for diseases, and many more. Introduction to types of the cloning. There are two types of the cloning: in vivo and the in vitro. Now, what is the difference between the in vivo and in vitro? In vivo involves the use of the restriction enzymes and ligases. Restriction enzymes for the cutting of the gene and ligases for the rejoining. You can say ligases act as a glue, using vectors and cloning the fragments into the host cell, like using the bacterial cell. And the other type is in vitro. In vitro, which is using the polymerase chain reaction (PCR) method to create copies of the fragments of DNA. Hundreds and thousands of the copies of the DNA can be produced by the PCR as well. This is the in vitro, and the other is in vivo, which is inside the host cell, like bacterial cell. So the concept clear about the in vivo and in vitro cloning. Now I'm going to discuss about the recombinant DNA. As you can see here on the screen, the recombinant DNA produced by the joining of the two different DNA, one gene of interest and other with the plasmid vector, which is a vector, and the rejoining of these two different genes give rise the recombinant DNA. So recombinant DNA is made by combining DNA from two or more species or sources. In practice, the process often involves combining the DNA of different organisms. The process depends upon the ability to cut and rejoin. Is it clear? So first one is the cutting of the desired gene and then rejoining of that into another. So this produces the recombinant DNA. Cutting of the gene carried out by the restriction enzymes, which are the endonucleases enzymes, and the rejoining of the two different genes or two different fragments of the DNA carried out by the ligases, which act as a glue. Is it clear? Okay. Now a little bit about the history. You can see here the Paul Berg on the left side and Stanley on the right side and Bohr in the middle. These are those scientists who worked on the recombinant DNA technology and first you can say that identified and discovered and made the recombinant DNA. And the first one, the biochemist at Stanford, who was among the first to produce a recombinant DNA molecule in 1972. And then later on, the Stanley and the Bohr developed techniques to make recombinant DNA a form of the artificial DNA. Is it clear? So here I'm to tribute to these legends who spend their lives for the science and technology and make the things easy for us. Okay, now recombinant DNA technology. It consists of the following steps, like the gene of interest, or you can say this process need gene of interest, molecular scissors. Molecular scissors means restriction enzymes, and molecular carriers, which are vectors, molecular glue like ligases, and expression system like bacteria. Is it clear? So I will discuss all these things one by one. Now you can see here, this is the whole mechanism of recombinant DNA technology. And just suppose this is the human cell, and from the human cell we want to clone any gene and make the recombinant DNA. Then what will happen? First of all, we take this gene by cutting with the help of the restriction enzyme. You must keep it in your mind, the restriction enzymes for the gene of interest, cutting for the gene of interest, and for cutting the vector which is used for that purpose, like this is the vector plasmid from the bacterial cell. Both are cut with the same enzyme, same restriction enzyme. If we use different enzymes for the cutting of the like the gene of interest and for the plasmid vector, then what will happen? Different restriction sites, these enzymes, different enzyme cut the DNA at different restriction sites. So it will not possible, or you can say that it will not produce the recombinant DNA. So what will happen? Same restriction enzymes and nucleases used for the cutting of the desired gene as well as the plasmid. So what will happen? So this desired gene, after cutting with the restriction enzyme, and this part of the plasmid also cut with the same restriction enzymes at the restriction sites, which I'm going to discuss, and then rejoining with the help of the ligases, and then after this produces this is the recombinant DNA. Okay, now this recombinant DNA put into the expression system, or you can say that a bacteria for the purpose of the cloning. As we are familiar that most Escherichia coli is used, which reproduce very rapidly. After 20 minutes, one bacteria give rise to bacteria. So the number of clones increases with the passage of the time. Okay. So first of all, here I'm going to give you some information about the restriction enzymes. What are restriction enzymes? They are also known as restriction endonucleases, are prokaryotic enzymes that recognize and cut DNA at specific sequences, and these sequences are known as restriction site. As you can see here, this is the restriction site in the bacterial plasmid or bacterial DNA, and this is the site, restriction sites in the chromosome or DNA of the human cell. So restriction sites are those sites at which the restriction enzyme cut the DNA. It is believed that they evolved as a defense mechanism. Now, what are restriction enzymes or endonucleases are the prokaryotic enzymes, and the bacteria have these enzymes as a defense mechanism for the foreign DNA like viral DNA. Over 3,000 restriction enzymes have been identified. Restriction enzymes are named based on the prokaryotic organism they are isolated from. Is it clear? Like if we are getting the restriction enzyme from the Escherichia coli, they are represented, or they are abbreviated as ECO, and many more I will discuss in detail in my next lecture about the restriction enzymes. Okay, as you can see here, this is the, just suppose this is bacteria, this is the gene of desired, and we are using the EcoR1. ECO means this restriction enzyme obtained from the Escherichia coli. So this EcoR1 cut the DNA at specific position, like at this site, at this site. These sites are known as in the DNA. This is the DNA. Just suppose this is the DNA, whole DNA, complete DNA. So what will happen? The restriction enzyme will cut the DNA at these sites, specific sites, and these sites are known as restriction sites. So you can see here the two fragments produced, and these two fragments are the sticky ends, and I will discuss what are the sticky ends and what are the blunt ends. So this is the sticky end. Is it clear? And now you can see here the difference between the other fragments. The fragment cut at this site. Is it clear? So after cutting, what will happen? Two equal size fragments are produced, and these fragments are known as blunt ends fragments. These are the sticky ends, and these sticky ends are most useful as compared to the blunt end. They easily reunite and useful for, you can say that producing for the recombinant DNA. Is it clear? So one is the sticky end producing and other is the blunt end, and these are the restriction enzymes which cut the DNA at specific site, which are known as restriction sites. Okay. Now, what is palindromic sequence? Most of the students are not familiar with the palindromic sequences and they're confused. Now, I'm going to resolve this issue. A palindromic sequence is a sequence made up of the nucleic acids within double helix of the DNA and RNA that is the same when read from 5 to 3 on one strand and 5 to 3 on the other strand. Must keep it in your mind. 5 to 3 end, the sequence will be same on both the strands of the DNA. Complementary strands. It is also known as palindromes or an inverted reverse sequence. Palindromic sequence is also known as inverted reverse sequence. Now I'm showing you here how. I'm giving you the clear concept of the palindromic sequence. Now you can say this is these are the two complementary strands of the DNA. If you read from this site, like this is a restriction site, these are the restriction sites. So if you read from this restriction site, you can see 5 to 3 TTAGCAC. And now on the other hand, you can also read from here that TTAGCAC. So both the sequences on both strands from 5 to 3 and are what? They are same. So these sequences are the palindromic sequences at which the restriction enzyme cut, and that sites are known as restriction sites. So palindromic sequence is the same backward and forward on both sides. This means that the enzyme recognizes the sequences no matter from which side the enzyme approaches the DNA. It is easy for the restriction enzyme to recognize that. Okay, so palindromic sequences also increases the chances that both strands of DNA are cut. Is it clear? Now, the concept about the palindromic sequence is clear now. So, now isolation of first step of the recombinant DNA technology is the isolation of the desired gene. At first, a donor DNA fragment should be isolated. There are two methods for isolation of desired gene or DNA fragment. Number one is the restriction endonucleases. As I have discussed, the restriction endonucleases cut the DNA at specific restriction site. And the other one is the using reverse transcriptase enzyme. Now, what is reverse transcriptase? Reverse transcriptase enzymes are those enzymes which synthesize the DNA from RNA. Is it clear? If you have no concept of it, must read about that. What are the restriction endonuclease, reverse transcriptase enzymes? So reverse transcriptase enzymes help in the formation of the DNA from RNA. If you have RNA, you can easily make your DNA from that RNA. So this mechanism is known as reverse transcription, and the enzymes are known as reverse transcriptase enzymes. So how many ways? Two ways. One is the restriction endonucleases ways, and other is the reverse transcriptase enzymes way. Okay. Now, selection of the suitable cloning vector. This is very important. First step is the isolation of the gene of desire. Second one is the selection of the suitable cloning vector. When the donor DNA fragment is incorporated into a host cell, it will not replicate because the isolated gene do not have the capacity to replicate itself. So before introduction of the donor fragment into the host, a suitable vector should be selected. Must keep it in your mind. Suitable vector before cloning should be selected. Cloning vector is the DNA molecule capable of self-replication inside the host cell, like the bacteria. The main function of the cloning vector is to replicate the inserted DNA fragment inside the host cell. Example of the cloning vectors are plasmid, bark, yak. I will discuss in detail in my next lecture about the vectors. So expression vector, etc. Characteristics of the good cloning vector. It must be self-replicating inside host cell. You can see here the structure of the cloning vector. And you can see the first one is the self-replicating, means it must have what? Replication site. It must have replication site. Or you can see the origin of replication. It must possess restriction sites for the RE enzyme. But must possess the restriction sites. Is it clear? Now, third one, introduction of the donor DNA fragment must not interfere with the replication property of the vector. Introduction of the donor DNA fragment must not interfere with the replication property of the vector. This is very necessary. This is very important. It must possess some marker gene, and marker genes are used for the selection of the particular clone from the thousands and millions of the bacterial colonies. Is it clear? So this selection marker, you can say that any antibiotic resistant gene. Is it clear? Okay. Now, incorporation of the donor DNA fragment with the plasmid vector. Now you can see here, this is the desired gene and this is the vector. After cutting both with the restriction enzyme, what will happen? Rejoining with the help of the ligase occur, and then this is known as the incorporation of the donor DNA into the vector, into the plasmid vector. The plasmid vector is cut open by the same restrictions I am not discussing here again and again. I have discussed in detail. Okay. Now, the transformation of the recombinant vector into the suitable host. The recombinant vector is transferred into the suitable host cell, that is bacterial cell. As you can see here, after producing the recombinant DNA, means the rejoining of the two different DNA, then this recombinant DNA put into the into the bacterial cell for multiplication. So this is known as the transformation of the recombinant vector into the suitable host. The recombinant vector is transferred into the suitable host cell, that is bacterial cell. Some bacteria are naturally transformable. They take up the recombinant vector automatically, like Bacillus, Haemophilus, Helicobacter pylori. These are naturally competent. They can easily take up the vector inside. But there are some other bacteria which are not naturally competent. In these bacteria, recombinant vector are incorporated by the artificial methods, such as by increasing the concentration of the calcium ion and by electroporation, etc. Is it clear? So this is how you can. Now, isolation of the recombinant cells. This is very important, the last step. What will happen? The recombinant host cell is then grown in a culture media, but the culture may contain colonies, both recombinant cells and non-recombinant cells. For isolation of the recombinant cells from the non-recombinant cell, marker genes of plasmid vector is employed. Like here you can see that this is the marker gene, which is ampicillin. This is the that gene which resists against the, or you can say that about the resist against what? Antibiotic. So this is the resistant gene which acts as a marker in the plasmid for the selection of the desired clone or you can say desired colonies of the bacteria from the number of colonies, non-recombinant colonies. So, for example, PBR322 plasmid vector contain different marker genes, ampicillin-resistant gene as well as tetracycline. And if we put ampicillin and tetracycline in the colony of the bacteria in which the recombinant and non-recombinant bacteria growing, then what will happen? Only the recombinant DNA containing bacteria will survive with the application of the ampicillin and tetracycline antibiotic. Other will die because the recombinant DNA contain the ampicillin gene, which is tetracycline gene, which is responsible for the resistance to the antibiotic. Okay. If knockout ampicillin resistant gene, what will happen? When PST1, this is another enzyme, restriction enzyme, is used, it knock out ampicillin resistant gene from the plasmid. So what will happen? So that the recombinant cell becomes sensitive to ampicillin and maybe die. Maybe. Okay. So this is the overall process which I have discussed. First the desired gene, then rejoining and making the recombinant DNA, and then this recombinant DNA put it into the expression cell, which is the bacterial cell, and from that it is selected by using the antibiotics. And this is the overall reaction. This is the overall process. I hope so the lecture will be clear upon you people. If you still have any question, kindly share with me. I will try my best to answer you. So thank you very much. I will be back. Till then, Allah Hafiz.